make people vulnerable to bacterial infections like tuberculosis, Lyme disease, and meningitis. So what's this got to do with language? Here, the 3D framework is useful. We know that bacterial diseases have mechanisms operating on at least two time frames. There's the moment-to-moment mechanism, which is the initial exposure to the bacteria, and there's also the evolutionary mechanism, which is the genetic susceptibility of the person exposed. Unless you understand both mechanisms on both time frames, you won't understand the disease. And this explains differences across species as well. There are some bacteria that infect only humans because only humans have a suitable and receptive genetic environment. Well, the same story applies to language. As we learned in the last lecture, being exposed to language is not enough to ensure its development. In this lecture, we'll explore the genetic and biological landscape that allows language to thrive in humans, but not other species. First, some basics on genetics. The latest estimates show that our bodies contain approximately 35 trillion human cells. I say human because it's only part of the total number of cells in our body. What about the other part? Well, largely bacteria. We'll come back to this in the next lecture. Almost all human cells contain a long coiled molecule of DNA that's organized according to 23 pairs of chromosomes. Each of these chromosomes contains hundreds or thousands of individual protein-making molecules called genes. Altogether, we have about 20,000 genes. These genes provide the basic instructions of what each of your cells is supposed to do. One rough analogy for understanding this genetic organization is a cookbook. DNA is the book, chromosomes are the chapters, and genes are the individual recipes. Most humans have the same basic book with the same number of chapters and pages, but some of our recipes are unique. This variation is caused by novel combinations of your parents' recipes. You inherit and combine most chromosomes from your mother and your father. And some of the variation is due to copying errors or genetic mutations. Within the human species, there's remarkable overlap of genetic material. We share about 99.5% of DNA with one another. And there are striking similarities across species too. You probably already know that we share much of our DNA with chimpanzees, about 98.5%. But you might be surprised that we also share 90% with cats, 70% with zebrafish, and even 50% with bananas. This highlights an important principle in evolution. Although there are genetic differences across species, there are many fundamental processes that are evolutionarily conserved. Evolutionary conservation means that basic genetic mechanisms are reused within a large number of species. Often, these are biological processes that serve basic survival functions, ranging from the very small, like ion channels that modulate cell activity, to the very large, like organs such as the eyes, heart, and lungs. This shared equipment is worth highlighting because it suggests that the unique things we see in particular species need not require completely new genes. This logic runs counter to most people's intuitions about language. Because language is so complex and so novel, it seems logical that there must be a specialized set of genes dedicated to it. This conclusion is understandable given that I've spent several lectures telling you how unique language is. This beguiling uniqueness has tempted many a scientist to make bold genetic claims about it. Perhaps the most famous in recent years is the claim made by the Harvard psychologist Steven Pinker in his popular 1994 book, The Language Instinct. In the book, Pinker builds on Chomsky's view of the uniqueness of language by putting an evolutionary twist on the original argument. In one well-known passage, Pinker draws an analogy to spiders. I'll paraphrase. Just like spiders spin spider webs because they have spider brains, humans speak language because they have human brains. Aside from being a bit circular, Pinker's logic assumes that because there is a radical species difference in a behavior, or a phenotype, the relevant genetic profile or genotype must also be radically different. The Language Instinct produced a scientific backlash, and since its publication, there's been mounting evidence for an alternative view of how language evolved. To appreciate this alternative perspective, we need to go back to our second lecture and recall that language is not a single thing, it's a system of things. Evolution may have selected for the system, but there's no doubt that parts of the system were borrowed from genetic material shared with other species. This is the basic idea of evolutionary conservation. Evolution often tinkers with old things to make new ones. As it pertains to language, this view is best summed up by the late great cognitive scientist Elizabeth Bates. Unlike Pinker and Chomsky, Bates did not see language as a new machine built out of new parts. Based on the principle of evolutionary conservation, Bates viewed language as a new machine built out of old parts. There are two main genetic arguments to support Bates's new machine, old parts view of language. The first is that there are no single genes for any complex behavior. Just like there will never be a gene for empathy, schizophrenia, or belief in God, there will never be a gene for language. And two, there's growing evidence from molecular biology that genes involved in language are not unique to humans. As with other cases of evolutionary conservation, the genes used in language are shared among many species. Putting these two things together, I now want to focus on one particular gene that has received the most attention in the molecular study of language. It's called the FOXP2 gene. This gene was the first to get scientists truly excited about understanding genetic mechanisms for something as complex and as unique as language. The story of the FOXP2 gene starts with a British family in the 1980s, medically dubbed the KE family. Members of this family were observed to have an unusually high preponderance of a particular disorder called developmental verbal dyspraxia. One of the defining symptoms of this disorder is a difficulty combining phonemes while saying words. For example, an extreme case would be someone trying to say the word brick, but regularly omitting the first few phonemes and just saying ick. Importantly, the muscles of the tongue and jaw are perfectly fine, so the breakdown is in how the brain orchestrates the speech movements. The disorder in the general population is quite rare, but across 3 generations of the KE family, about 50% of the members had some form of it. This led to the hypothesis that there may be a genetic mechanism underlying the problem. It took a while for the tools of molecular biology to advance enough to confirm this hypothesis. But in 1998, neuroscientist Farina Varga-Kerem, geneticist Simon Fisher, and their colleagues published a paper locating the problem on a stretch of genes on chromosome 7. Later, in 2001, using more powerful gene imaging techniques, the exact location of the mutation was identified on the FOXP2 gene. Later still, it was discovered that the problem was caused by a modification to just a single letter of the FOXP2 DNA sequence, which was traced back to a mutation passed down by the grandmother of the KE family. Although the FOXP2 was initially celebrated by the press as a language gene, the scientific community was much more cautious. And with good reason. It turns out that the FOXP2 is one of the most evolutionarily conserved genes in all of the animal species. Not only do we have it, but so do chimps and birds and even bees and alligators. Now, even though it's shared across many species, there are small but important differences. To appreciate the differences, you need to understand how genes operate. Genes churn out gene products, such as RNA and proteins, that act on other cells. These gene products are the real heavy lifters in making us who we are. The FOXP2 gene produces a protein creatively called the FOXP2 protein. The human version of this protein differs by only 2 out of 175 amino acids from the chimp version, and by only 3 from the mouse version. This protein acts on various other cells during prenatal development to affect not just the brain, but also other organs, such as the heart and the lungs. Although the FOXP2 gene is implicated in many aspects of an organism's development, which is another good reason not to call it a language gene, its effects on the brain are directly connected to language. In humans, the brain regions that are affected downstream by the FOXP2 mutation are areas involved in motor control of the mouth and tongue, which are clearly necessary for speaking. One of the major lines of evidence to support this comes from research on non-human animals. For example, Constance Scharff at the Free University of Berlin found that disrupting the FOXP2 gene in songbirds caused downstream problems in a region of the brain called Area X that controls song production in birds. Going a step further, researchers are using genetic engineering to test the role that human genes play in non-human animals. Using state-of-the-art gene editing techniques, scientists now have the capability to extract segments of human genes and insert them in the genome of other animals. For example, Wolfgang Einard at the Max Planck Institute for Evolutionary Anthropology in Germany has done research that snips the human mutation of FOXP2 and places it in the appropriate segment of the genome of living mice. The result is a significant disruption to motor brain regions involved in controlling high-pitched vocalizations, which is how baby mice communicate with their mothers. Einard also describes the opposite technique, inserting a fully functioning human FOXP2 gene into a mouse genome and testing what happens. Remarkably, this resulted in a proliferation of neural connectivity in motor regions in the mouse's brain. The research is ongoing, but if these changes correlate with enhanced vocal performance in the genetically altered mice, it would suggest that the human variant of the FOXP2 gene may have indeed been an evolutionary springboard for more simple communication to the complexities of language. Another astonishing development in studying genetic mutations for language comes from the field called molecular anthropology. You may know this field from the popular 1993 film Jurassic Park. The field has actually been around since the 1960s, but recent advances in gene sequencing are turning science fiction into reality. One of the leaders of the field is Svante Pääbo, also at the Max Planck Institute of Evolutionary Anthropology. He and his team are trying to trace the modern human genome to our ancient ancestors and even closely related cousins that have become extinct. Using DNA from 50,000 years